All right, I am going to go through the review. I'm going to cut out some questions because really with this unit, I see, I just see that I gave you information that I have always given students, but I'm starting to see the trend from College Board that some of this stuff they're just not going to ask about. But it is good to be exposed to it, but I don't need you to study some of it for the test. So I'm looking at the test review. Number one definitely is a good one to know. Asexual versus sexual reproduction. Asexual reproduction is advantageous because it is fast and energy efficient. And if you have an organism that is genetically, I don't know, I would say like perfect for the environment or has really good fitness for the environment that they live in, asexual reproduction is perfect, is fine for that organism because it would produce clones of it. Sexual reproduction is slower and energy consumptive, but it leads to a lot of variation and diversity in the populations, which is the foundation for natural selection and evolution. Crossed out the one about parthenogenesis. That was just supposed to be like an interesting FYI. You don't need to know that for your test. You don't need to know that. Oh, differences between somatic cells and gametes. A somatic cell is a body cell that is diploid. A gamete is an egg or a sperm, and it's haploid. This is not in very good order right here. So it should say somatic cell, then gamete, then autosome, then sex chromosome. So I'm sorry, that's not in a very good order. So somatic cells and gametes are the types of cells. Somatic cell is a body cell, gametes a sex cell. And then the two words in the middle, autosomes and sex chromosomes, those are the types of chromosomes that humans have. The sex chromosomes that humans have could be an XX or XY. And then anything that is not an X or a Y is called an autosome. So remember, chromosomes are what DNA consists of DNA and the proteins that it's wrapped around, and it is where your genes are held or stored. Number three, what are homologous chromosomes and sister chromatids? So a homologous chromosome is going to be a type of chromosome where you have one from mom and one from dad. So here's a pair. If you look at this shot I have up here, this would be like an example would be, this is chromosome number 1. The copy of chromosome 1 that you have from your dad is here. The copies of chromosome number 1 you have from mom is here. Together they make up homologous chromosomes. So you have 23 sets of homologous chromosomes as a human. Whenever the chromosome copies and duplicates, those are now called sister chromatids. Sister chromatids. So those are the differences. I already went over sex chromosome, autosome, haploid, diploid. So we're going to go to number 5, the steps of mitosis, or meiosis, meiosis. All right. So just in general, prophase is the condensing of chromosomes, and that is when crossing over occurs. Crossing over is just when a little piece of one chromosome switches with another piece of another chromosome that's next to it. They're all homologous, and so that just kind of mixes things up. That leads to genetic variation. In metaphase 1, the tetrads line up, so homologous chromosomes line up. The homologous chromosomes and their copy line up in metaphase 1. In anaphase 1, the homologous chromosome separates, and then telophase cytokinesis, basically you end up with 2 cells at the end. Then those 2 cells, so you're seeing them right here, so what I just described is in this picture, those 2 cells go through another division. There's no interphase, there's no crossing over in the second division. The spindle just reforms. Sister chromatids that have been kind of mixed up because of crossing over then line up on the metaphase plate. Anaphase, sister chromatids separate. Telophase cytokinesis, now you end up with 4 genetically unique haploid cells, the sperm and the egg. All right, make a sentence, this is number 6. Make a sentence using the following words: crossing over, synapsis, chiasmata. Well, I'm going to go back to the crossing over. Okay. let's see. Homologous chromosomes pair up. I'm having trouble making this sentence. Okay. Well, synapsis is when homologous chromosomes pair up during prophase and they perform crossing over at the site. The site of crossing over is the chiasmata. So get from that what you will. I'm going to keep going. Okay. 7, what happens at the end of meiosis 1? What's the result? The result of meiosis 1 is right here. 2 haploid cells form. Each chromosome still consists of 2 sister chromatids. So that's the answer. So pause if you need to. What happens at the end of meiosis 2? It says right here. You have haploid daughter cells that are genetically unique. So 4 haploid daughter cells form, and they're genetically unique. When, I'm on number 9, when during meiosis are homologous chromosomes separated? The answer is metaphase 1. When during meiosis are sister chromatids separated? The answer is metaphase 2. Number 11, what does the human life cycle look like? Let's find this. Okay, here's this cool picture right here. You're just looking at this picture on page 173. Okay, so you have the ovaries and the testes, and you have a man and a woman. Inside the ovaries, meiosis occurs and you form eggs. Inside the testes, meiosis occurs and sperm are formed. When a sperm and an egg fuse, that's called fertilization. So if you have a haploid and a haploid fusing, that forms a diploid, and that's called a zygote. And then the zygote is going to multiply by the process of meiosis, or the cells are going to divide, and by the process of mitosis, and then that's how it develops into a large organism. Cool. I think I said all those words. Yay. Number 12, what are the sources of genetic variation in a sexually reproducing organism? Number 1, crossing over. Number 2, independent assortment of chromosomes. And number 3, random fertilization. The fourth one would be mutations, but that doesn't have anything to do with meiosis and like sexual reproduction, so that usually isn't included in that, but just know that mutations actually do give genetic variation. All right, number 13, what are the differences between mitosis and meiosis? Mitosis has 1 division, meiosis has 2. Mitosis results in 2 diploid cells. Meiosis results in 4 haploid cells. Mitosis forms new cells that are clones of each other. And meiosis forms 4 genetically unique cells. Mitosis is going to involve the division of somatic cells, and meiosis is going to form gametes. There you go. Those are the differences, and they're on page 174. Okay, number 14, describe how human chromosomes are arranged on a karyotype. Well, they're numbered 1 through 22, and then the sex chromosomes, so they're just paired up. Homologous chromosomes are paired up, and they're arranged from like biggest to smallest. And then you have your 2 sex chromosomes at the very end. So that's what a karyotype looks like. What is non-disjunction and what does it result in? Non-disjunction. Non-disjunction is a failure for chromosomes to properly separate. During anaphase of meiosis, it results in either extra or missing chromosomes. Next page. Give an example of a non-disjunction disorder and the effects on the person. So Down syndrome is an example of a non-disjunction disorder. So that extra chromosome number 21 was a result of non-disjunction. Sorry, I got distracted. Yeah, so there's an extra chromosome 21, and that causes Down syndrome. All right, number 17. Oh my gosh, that means I'm on another chapter. Where's my other chapter? Hold on, let me see if we can find it. No. Genetics, AP Bio. Oh my god. Genetics. Ooh, I bet it's here. Yes. Number, hold on, chapter 11, Holbrook. I bet you it's not going to open. Oh, pause. Let me pause. Hold up. All right, here it is. I'm going to try to go down here. So I am now on number, what am I on? Number 17. What did Mendel discover? Well, he really discovered this dominant and recessive idea. So the Mendel's principles that you have alternate versions of genes called alleles that cause variation. For each character, every organism inherits one allele from each parent. If the dominant allele is present, it will be fully expressed. The recessive allele will have no noticeable effects on the offspring's appearance if the dominant allele is present. That's really it. He studied pea plants. It's important to know P generation, F1 and F2. So the P generation is true breeding. The F1 generation is the hybrid of the P generation. And the F2 generation is the hybrid. It's basically not the hybrid, but it's the just the offspring of taking two of these F1s and basically crossing them. Okay. I just did. Allele is an alternate version of a gene. Dominant, recessive, I just went over that. Homozygous, heterozygous. Homozygous is when both letters are the same. Heterozygous is when they're both different. Phenotype, genotype. Phenotype is the physical trait that is seen. Genotype is the genetic makeup, like the actual letters. And then a test cross can be performed if you have something that is expressing a dominant trait and you're not sure if it's homozygous dominant or if it's heterozygous. You can cross it with a recessive, and then you can tell from the offspring whether or not that original thing that you're trying to figure out is if it's homozygous or heterozygous. All right. Practice problems. I do not have, okay, I can try to do them here. I'm just going to talk through the problem. I'm not going to be able to use my pen. I don't have my little pen thing. So I'm just going to tell you what the answers would be. So number 19, in seals, the gene for the length of whiskers has two alleles. Long whiskers are dominant to short. What percentage of offspring would be heterozygous for long whiskers if a homozygous dominant long whiskered seal is crossed with a heterozygous long whiskered seal? So homozygous dominant. So you would do big H, cross it with a big H, little h. Big H, big H, big H, little h. And it wants to know how many would be heterozygous. It would be 50%. Yeah, 50%. How do you solve a dihybrid cross if both parents are heterozygous for both traits? You would do the 9331. And if you're confused on that, you can look on page, hold on. Well, it's in the notes. Just find the 9331 thing in the notes. I'm not going to be able to find the page number this second. Okay, number 21, how would you solve a dihybrid cross that has one parent heterozygous for both traits and another parent homozygous dominant for both traits? Ooh, this one's going to need to be worked out. So you have a parent heterozygous for both traits. That means that all four alleles are possible. So you'd have all four gametes, and then the one that is homozygous dominant for both traits, you would just have one gamete. So this is a 4-box Punnett square. So that's how you'd solve that one. Give an example that shows incomplete dominance. Go to page 182. You could have a red flower and a white flower to make pink flowers. Number 23, give an example that shows co-dominance on page 183. You could have a white horse and a red horse, and they will make roan horses. Or you can have AB blood type. That's also co-dominance. Give an example cross that shows blood typing. Just look on page 184. Epistasis. This is number 25. That just means that a second gene is going to control the expression of a first one. So if the second gene in Labrador retrievers, when we talked about fur color, if the second gene was recessive, like homozygous recessive, then they're not going to show color in their fur, basically. And then what is pleiotropy? Pleiotropy is one gene is going to have an effect on multiple traits. polygenic inheritance. This is number 27. Sorry, I should be doing this. I should have been showing y'all. Oops, my bad. Whatever, I can't redo it. Polygenic inheritance, right here. That would be multiple genes affect one trait, like skin color and height. What did you learn about the color of a hydrangea flower? Hydrangea? Hydrangea flowers can vary in color depending on the environment. So it depends on the acidity and the aluminum content of the soil. So sometimes gene expression can be controlled by the environment. Number 29, in the pedigree below, could the pattern of inheritance be? So it's number 29 on the review. Autosomal recessive, autosomal dominant, X-linked recessive. So could it be autosomal recessive? You have parents, so you have individuals 1 and 2, and neither of them are shaded. And they have some shaded and some not shaded children. So yes, that could be autosomal recessive. Could it be autosomal dominant? No. Autosomal dominant traits cannot be hidden, and parents 1 and 2 are not shaded, but they have a child that is, so it couldn't be a dominant. X-linked recessive, yes, that could be X-linked recessive because parents 1 and 2, neither are affected, and then you have a disproportionate number of males affected. It's only males that are affected in this pedigree, so yes, it could be X-linked recessive. Cool. That's it for that one. Let me find my other slideshow. I hope it opens. Hold on, I'm gonna pause. Okay, this is chapter 12. I'm on number 30. Okay, wild type and mutant. Wild type means dominant. I'm on number 30, yes. Wild type means dominant. Mutant means recessive. Give an example of a sex-linked trait. That's number 31. Color blindness is a sex-linked trait or disorder. Hemophilia. Eye color in fruit flies. Number 32, show a cross between a color-blind carrier mother and a normal dad. So if you have a mother who's a carrier and a normal dad, none of the daughters will be affected. Half of the sons will be affected. Okay, we have an example of one of those Punnett squares in the binder. All right, number 33. What does the pedigree show on the bottom of page 197? Oh my gosh. Do I have to really look? Okay, page 197. Hold on, let me find it. Where is it? Page 197. Ooh, I don't even have it. Oh my gosh. I'm sorry, I don't feel like pausing. I'm just gonna have to wait a second. 198, please be 190, yes. Oh, okay, on the bottom of page 197, you see this royal family. You see that Prince Albert and Queen Victoria, neither of them have the disorder, but then all you see are some of the sons, but it's only males that are affected. And that's for hemophilia. So that was that big old pedigree that you saw on page 197. So, yep, mostly males are affected, X-linked recessive. Number 34, what is X inactivation? How does it apply to calico cats? That's not on your test, but in case you were wondering. Okay, in case you were wondering, 2 X's in a cell, like even in humans, one of the X's gets inactivated. It's only 1 X is working at a time, but it's random. So in cats, they could have an X for orange and an X for black fur color. Depending on which X's are inactivated in each cell, they have like splotchy, calico, tortoiseshell fur color. What is the SRY gene? It is the gene on the Y chromosome that codes for the sex characteristics of males. Number 36. There are like 3 or 4 questions on this. That's a lot. Out of 23, that's a lot. How can you tell if genes are linked on the same chromosome? If genes are linked on the same chromosome, you're going to see a situation like this, where there should have been an equal amount of each outcome, but there is significantly more parental types than there are recombinants. Therefore, the genes are linked on the same chromosome. Number 37, what accounts for the recombinance? So if the genes are linked on the same chromosome and they're inherited together, how do you even get recombinance? That's because there is a chance, depending on how close or far apart those two genes are on the same chromosome, that they can be separated from each other during crossing over. One map unit on a linkage map, I'm on number 38, means 1% recombination frequency. And it can give you a relative distance between two genes that are linked. Number 39, what does a 50% recombination frequency indicate? It indicates that the genes are either on complete opposite ends of a chromosome or they are on two completely different chromosomes. Number 40, what is genomic imprinting? That's actually not on your test. But just so you know, it's when a gene is inherited from the mother, it will be expressed differently than if it's inherited from the father. And that's because in the mother's egg, methylation can occur. All right. Number 41, which parent do you inherit mitochondrial DNA from? It's your mother. Number 42, what is the difference between aneuploidy and polyploidy? Don't freak out. I didn't put that on the list. Aneuploidy. Aneuploidy is just an abnormal number of chromosomes, period. So it could be like an extra copy of chromosome 21 or a missing copy of chromosome X. Polyploidy is a extra copy of every chromosome. So it's like two or more complete sets of chromosomes. I remember polyploidy happens a lot in plants. That's not on your test though. Number 43, what are the 4 types of chromosomal mutations? Deletion, duplication, inversion, and translocation. That's it. All right. Then it gives this long vocabulary list. So what I'm going to do is go through and I don't know what you're going to look at. You're just going to look at a black. Oh, there is chi-squared on there. Do chi-squared. There's chi-squared. I'm just gonna have a black screen, my bad. But I'm gonna go through and just really highlight and emphasize some things. First off, I want to emphasize the sources of genetic variation that occurs in sexually reproducing organisms. That's really important to know, especially crossing over. And so be sure that you would be able to recognize crossing over, like a picture that has homologous chromosomes performing crossing over. So go back and make sure you look at a picture of crossing over happening. Like not just knowing crossing over, but what visual is there for crossing over? And crossing over results in genetic variation. All right. There are 2 Punnett squares on dwarfism. You had a dwarfism problem. Go look back at your genetics problems. You have, I think, 3 dihybrid crosses. Go look back at those dihybrid cross problems that you did. Did I repeat this question? Oh, no, I didn't. No, I didn't. Okay. You have 2 pedigrees. I'll show you a pedigree and it'll say, based on this pedigree, predict the probability that the individual marked by the question mark will have the skin disorder. So it's going to take doing some Punnett squares. So if you're not doing a Punnett square with your pedigree and you're having trouble with it, do the Punnett square with the pedigree. On the second Punnett square, based on the data in figure 1, which of the following best describes the inheritance pattern? So look at the pedigree and tell me, is it autosomal recessive, autosomal dominant, X-linked recessive, or X-linked dominant, right? I would look at the similarity, the differences between mitosis and meiosis. There's 2 questions about that. What is the difference between metaphase 1 and metaphase 2? There's a question about that. There are 2 chi-squared multiple choices. One of them is going to ask you for the chi-squared value. And I think one of them is going to ask you, do you accept or reject the null hypothesis? I got to look at it. Hold on, I'm not there yet. You need to know what recombination frequency means. Go look back at that. We got another dihybrid cross. What proportion of the offspring are expected to have ebony bodies and sepia eyes? So a dihybrid cross with flies. Flies are so gross, but we talk about them so much in this class. It's weird. There's a question about non-disjunction. Do you know what non-disjunction is? There is an X-linked trait, hemophilia Punnett square. Ooh, another question about crossing over. What is crossing over? What does it look like? What's an illustration of crossing over look like? I have a question over the steps of meiosis. I'm going to give you 4 steps that occur in meiosis. You're going to put them in order. You need to know what a haploid cell and a diploid cell is. What does that mean? Yeah, differences between mitosis and meiosis again. Yep, there's the chi-squared question. Do you reject or fail to reject the null hypothesis? Great. Ooh, can you recognize when 2 genes are linked on the same chromosome given a data table? Can you recognize it? All right, what is this? Just a basic Punnett square. Just look over an example of a complete dominance, incomplete dominance, and codominance. Look at an example of a blood typing Punnett square. And then, yeah, that's it. I'm going to give you a data table for your FRQ. 4 crosses using fruit flies. You got to tell me the pattern of inheritance, the genotypes of some of the parents, why are all the offspring affected in cross 4? Okay. Yeah, it shouldn't be. This isn't a bad FRQ, I don't think, at all. Okay. Those are some really pretty solid hints. So if you're watching the video, then good job. I think you'll do fine. Just study and listen to this video again because I told you everything basically. All right, I'll see you guys later. Thanks, bye.